The urease test. The urease test is a simple broth inoculation. You will be inoculating 2 different urease broths. Be sure to label the tubes with the name of the bacteria prior to inoculation. Urea is a nitrogenous waste product produced when amino acids are broken down. It is a very rich nitrogen source. Some bacteria are capable of producing an enzyme known as urease. Urease will break down urea into water, carbon dioxide, and ammonia. The urease broth contains urea, a phosphate buffer, and the pH indicator phenol red. It is used to differentiate between rapid and slow urease producers. Rapid urease producers will break down urea and produce enough alkaline waste products to overcome the phosphate buffer in a 24-hour period. This will cause the pH indicator phenol red to turn a bright pink. Slow urease producers are not necessarily non-producers. They simply do not produce enough alkaline waste product to overcome the phosphate buffer in a 24-hour period. Many enterics are urease producers, they are simply slow urease producers. Slow urease producers are read as negative results in a urease test. When inoculating your tubes, be sure to use aseptic technique and place them in the incubator for 24 hours and no longer than 48. Results are determined by the observation of a color change in the media when the tubes are taken out of the incubator. There is no reagent required for interpreting results in a urease test. Interpretation of the urease test results. On the left is a negative urease and on the right is a positive. The organism on the right was capable of breaking down urea and producing enough alkaline waste products in a 24-hour period to overcome the phosphate buffer in the media. Now shown is a negative urease on the left, a positive urease in the middle, and a weak positive on the right. There are a few organisms that are capable of breaking down urea rapid enough to begin overcoming the buffer. However, they will not produce enough alkaline conditions to completely overcome the buffer and cause the phenol red to turn the positive bright pink. These organisms fall somewhere in between rapid and slow urease production. Urease production is found primarily in gram-negative enteric bacteria. Many of those enterics are known causative agents of urinary tract infections, since urea is the primary nitrogenous waste found in urine. The urease test is also used to differentiate Proteus, which is a rapid urease producer, from other gram-negative enteric organisms.